Morning, everyone, and welcome. We're going to start right on time at 11 a.m. here. And the prerequisite wait 5 minutes for people to join the call of the meeting will happen while I say a few words here. So welcome to today's event, Applications, Challenges, and Opportunities for Top-Down, Intact, and Native Mass Spectrometry and Biopharmaceutical Characterization, hosted by the Consortium for Top-Down Proteomics. I'm Jared Auclair, the Dean of the College of Professional Studies and Director of Bioinnovation at Northeastern University in Boston, Massachusetts. Just briefly, a mass spectrometrist by training, and now spend much of my time thinking about life cycle management of products, um, large molecule products. So, just to start, a huge thank you to our annual sponsors at the consortium, at the major sponsor level, Thermo Fisher, Bruker, SciEx, and Lilly. And then at the supporter level, Agilent, Pfizer, Nautilus, Impro, and New Objective. And without their support, we couldn't host events like today. We have a great lineup of speakers that we'll hear from throughout the next 3 hours. I will moderate and keep them to time. If there is time to answer their questions or to ask questions at the end of their presentation, you can do that by unmuting and asking the question. Raising your hand, unmuting and asking the question. I will call on you. You can also put your question in the chat and I can read it to them for you. If there isn't time for questions at the end of each presentation, at the end of the 3 hours as a just under 1 hour panel discussion where we will open the floor directly to you all, the participants, to ask questions of the panel, which hopefully will be all of our speakers or the majority of them, as opposed to me asking them canned questions. So you can hold them till then if you don't have the opportunity after their presentation. The agenda will be in the chat if it's not there already, but you can see briefly here what the agenda looks like for the next 3 hours really quickly. So nobody came here to listen to me talk. So without further ado, I'm going to stop sharing my screen and let Jack share his screen. And as he's doing that, we'll have Jack Kellie speak first. He'll provide a brief introduction of himself so that I don't take his time. And he'll talk about intact mass for ADC bioanalysis and practice, PK, DAR, and payload measurements. Jack, the floor is yours, and you have until 11:20.
Thanks, Jared. It's my screen coming
Yeah,
through. All
looks
right,
good.
with my title
Looks
slide there. Perfect. So my name's John Kellie. I go by Jack. I run a preclinical mass spectrometry bioanalysis group at AstraZeneca. We work with lots of different complex biomolecules. I'll touch on the ADC piece today, and with such a short amount of time, it will go quickly. So for there may be questions, I understand. So hopefully we'll have a few minutes at the end of the talk and then as well the panel time. So we'll go ahead and let's see, advance here. So as I mentioned, we're in the preclinical space. Let me see if I can get my laser pointer to go. Looks good. So essentially, once it gets ready to go to the clinic is when it's handed off from my group. But we'll handle all of essentially the pharmacokinetic and pharmacodynamic and safety studies will be handled by my team globally. So for ADCs, we have endpoints, which we'll go into a little bit more detail about, called total antibody, total ADC, which is conjugated payload. We look at intact and reduced ADC, which will be a highlight of the slides here. And then free payload, which I'll touch on briefly, but that's small molecule and probably of less interest for this group, understandably. One note for nomenclature, so I'll refer to stochastic and site-specific ADCs throughout the talk. So a site-specific ADC is engineered for one specific position, such as an unnatural amino acid, where we can attach the linker and payload. A stochastic ADC is conjugation to the cysteines. And we'll touch on it, but essentially the site specifics we can look at by complete intact mass. Stochastic ADCs, we would typically reduce to the heavy chain and light chain because the interchain disulfides are broken to conjugate on the linker payload. So we'll talk about the basics of these total antibody and total ADC measurements. I'll talk about how we do the intact and reduced ADCs. One slide on conjugated payload, and then we'll move into the case studies, which is where the kind of the cool data will sit. Important to kind of set up context here, what we're doing. So we're looking at these preclinical in-life studies. So these are rats, mice, monkeys dosed with our ADC. We collect plasma or tissue, depending on, for example, if we're looking in blood or tumor. And then we'll assay that. So all the data I'll show here is from plasma. So we'll take, for a single analysis, usually between 20 and 50 microliters of plasma, perform immunocapture via an anti-human IgG capture reagent, perform for total antibody and total ADC, reduction, alkylation, digestion. And then from there, we split the sample essentially and go on to quantitate a surrogate peptide via triple quad mass spec, or we can do an enzymatic release. So these linkers are known to be cleaved by certain enzymes. So another enzymatic reaction, release the payload, and we know from the immunocapture step this is attached to the antibody. Essentially, also triple quad MRM type measurement. For the intact and reduced, it's pretty much the same workflow, except we don't do the digestion steps. We'll just take that sample, throw it directly on the mass spectrometer, whether intact or reduced. For the site specifics, we'll get some certain DAR profile. It's pretty straightforward. For the reduced, you have heavy chain and light chain peaks, and then depending on how it's conjugated, you may have some payload on the light chain, some on the heavy chain, and then you have to do some math to correlate back to total DAR, but essentially it is pretty straightforward. And it's important to look for free payload as well. I'm not really going to talk about it here, but there are lots of small molecule analysis strategies, such as protein precipitation and solid phase extraction. And of course, when you precipitate out proteins, you remove the actual ADC molecule. So it's pretty straightforward. And looking at free payload can be really important for safety or to understand DAR loss in terms of essentially the exposure to the free payload. So into the case studies here as I move quickly along. So this will be comparing some digestion and reduced ADC data for a non-human primate study. So in the world of bioanalysis, we deal with calibration curves and known QC samples. And so this is just an example of a calibration curve. A curve is spiked into plasma over a certain range. In this case, it's something like 0.1 to 50 micrograms per mil. And then we can back calculate from the response and fit unknown samples to this curve. So when we fit the unknown samples to a standard calibration curve, then we get PK results. So here are a couple different doses, total antibody and total ADC. And you can see the total ADC starts to become lower than the total antibody. So this means some deconjugation is occurring. So now at the digestion level, somewhat analogous to the proteomics world, we can see the pieces of the larger molecule and we can infer what is happening, but we don't know exactly what is happening unless we look at the intact mass. So that's the next step here. So looking at the reduced, in this case, ADC, allows us to see what exactly is going on in terms of why we're observing essentially a loss of linker payload or a lower concentration of conjugated ADC than we have a concentration of total antibody. And here, so this is cysteine conjugated, so it's by reduced. So these malamides can undergo retromical, and that's kind of what we see here. So we have a loss of just the linker payload. So it's pretty clear over time we're going on the heavy chain, the light chain, respectively, from the expected number of linker payloads, losing an entire linker payload unit, another entire linker payload unit, and it's pretty explanatory as to the mechanism for DAR loss. And here we compare the data. So we've got digestion data in red, our reduced mass data in blue. And when we normalize everything to sort of percent DAR, ratio of percent DAR remaining as being 1 or 100%, then we can take a look at how this data is similar. And in this case, they correlate very well. The digestion data compared to the reduced data. And so while everything doesn't always line up nicely like this, data, as shown here, help give us confidence, not only from a proof of concept, but just from a practical passing the eyeball test, that yes, these intact and reduced methods can help give us the same quantitative responses as digestion-based methods. And this gives us confidence to be able to deploy both depending on the opportunity and the situation. And so the situation then kind of being, how do we leverage an intact measurement to learn the most about the molecule and get a lot of data quickly? So that's kind of then as I'll jump into point number 2. So this one's going to be site-specific here. So only intact makes it a little simpler. And that method is the same as I showed a few slides ago, but I really just wanted to highlight the fact that we're dealing with now a completely intact ADC, mass around 150 kilodaltons, and ideally a pretty simplified DAR distribution. Now, here, this is a site-specific ADC, so there's no chance for the retromichael, but we can still understand why we might see DAR loss. And so first I'll talk about the individual biotransformation examples and why an intact approach is not only useful, but if you're like me, you think it's pretty cool. So essentially, this one's a site-specific DAR4, and there's some DAR3 and some DAR5. And initial time point, everything looks fine, but 72 hours down to 1 week, we start to see some other peaks. So this is a loss of about 1,100 daltons. And of course, I can't share the structure because it's proprietary, but essentially, the red part is the warhead. And there's a certain chemical structure that would mean losing this piece is about 1,100 daltons. So it's a really good way to identify the exact nature of deconjugation, or rather biotransformation on the payload. And in fact, from this set of studies, which kind of had different similar types of molecules, as you will see in the next few slides, but slightly different structures. In some samples, we could see loss of a mass unit of about 250 kilodaltons, which essentially correlates to a functional subunit here. So rather not loss of the whole piece, but of the an individual subunit. And we can see from loss of the DAR2, we can see it being removed from both of the linker payload species. So very useful, very cool for us to be able to tell discovery teams exactly how the molecule is falling apart, if indeed the ADCs appear to be unstable. But certainly what we want to do is we want to be able to create stable ADCs. And so this might mean screening lots of different molecules with different conjugation sites, different DAR ratios, maybe slightly different payload structures as the medicinal chemists will come in and work to optimize that payload molecule. So here we dosed 20 different ADCs. We have our PK time points, and we did all of the PK and DAR in this case by intact mass with no digestion, really going for speed and simplified measurements and an abbreviated calibration curve. For digestion-based and advanced PK measurements, it's more than 4 points, but for here, we're screening a lot of molecules quickly. So just looking at the intact mass response with respect to concentration, and to get a peek at the data from some of the molecules, we can see intact mass DAR2, DAR4, in theory, there should be DAR2 and DAR4, some slightly different DAR, 1.8, 1.82, 2.14, but close enough to 2. Similarly, 4.4, 4.3, close enough to 4. So be able to quickly determine essentially both PK concentration based on a curve like this and percent DAR remaining can be pretty useful. And here it is put through with the data that comes out. So we've got grouped by DAR2 and DAR4 ADCs. And essentially, this y-axis here is percent DAR loss. So some of the stable molecules are within sort of marked to 20% by day 7 threshold as kind of our stability, I guess our stability point where we consider it stable versus not stable enough to continue on down into future studies. So a few of these high flyers we can see are really unstable. Interestingly, the DAR4s had much more stability compared to the DAR2s for looking at just the overall percent DAR loss. So essentially, we're able to quickly screen and look at these molecules. But because it was an intact PK measurement, kind of the challenge was to really get the best PK data and DAR data. And so this is kind of the best of both worlds aspect, which is how this is actually how we're actually doing it now. So we don't quite have all the data because a lot of this I can't share because it's proprietary. But essentially, we do the digestion-based workflow, look for the green light, and then that green light, what is that? Well, it's enough drug on board for me to see the intact molecule. The intact measurements, as we know, are always going to be less sensitive than peptide measurements. Maybe not. Maybe I'll be proven wrong in the session here, but certainly in my hands, intact protein assays are much less sensitive than digestion-based. So we get a good measurement of the PK and a better estimate of molecule concentration than we can get from intact. And then we come on with the intact workflow to look at essentially DAR and conjugated ADC. And our current limits of detection, we're working to get these lower, but around a microgram per mil in plasma and serum. And with more DAR species, our sensitivity, of course, becomes worse because there's more peaks to deal with. And that's kind of how we're tackling things now. So I think I'm okay on time. So I'll just throw up the acknowledgement slide. And thank you all for listening today.
Check. Wonderful. If you have any questions, if there are any questions, please go ahead and raise your hand. You were spot on time. While people are thinking, I'll ask the sort of out of left field question that's more up in the air, perhaps. But as I think about where industry and therapeutics are going, there's much more talk about AXCs and PXCs, so antibody, whatever, conjugate, oligo, or whatever, and same thing with proteins. I'm just curious from your perspective if you see any analytical challenges as those molecules evolve based on your expertise and experience with ADCs.
So, I mean, sure, but the overall strategy, the strategies between here and here can be pretty straightforward. For example, an antibody oligo conjugate, maybe the properties of the oligo make the intact measurement harder, but the reduced is okay. Or maybe we have to somehow cleave off that oligo and do mass spec. Or if we can't do mass spec, do PCR. For an antibody peptide conjugate, it's pretty simple because it's just another piece of a protein. And like small molecules, anything that you're going to conjugate to an antibody has the risk to change its stability, its hydrophobicity, its PK. So, I mean, analytical challenges, sure, but that's our job as analysts in pharma is to solve these problems and do it quickly.
Agreed. We like to solve them too in academia, but there's fewer of us than there are of you in industry. Well, thanks, Jack. That brings us up on time. So we'll move on now to hear from Lucile, who's going to talk about state-of-the-art characterization of in vivo biotransformations of biotherapeutics. on the new Tim's Omni platform. Lucille, the floor is yours, and you have until 11:35. Your slides, but I cannot hear you.
Sorry. I said thank you for the introduction. I'm Lucile Kogey-Fuchs, a third year PhD student, and I'm doing my PhD in collaboration between Sanofi and l'Institut Pasteur. And today I will present my recent work regarding the characterization of in vivo biotransformation of Biotherapeutics on the new Tim's Omni instrument. A little bit of context. As you all know, antibodies have revolutionized targeted therapy by enabling highly specific recognition of disease-associated targets. This specificity comes from the complementary determining region, which define antigen recognition and binding specificity. You can find them in the Fab region. Today, antibodies form the backbone of biologic therapies with more than 200 antibody-based biotherapeutics on the market by 2025. These molecules are already complex biological constructs, but we are now witnessing an emergence of even more sophisticated formats known as next-generation biotherapeutics. These include bispecific and trispecific antibodies that are designed to engage multiple targets simultaneously, therefore improving efficacy. With this increase in diversity and complexity, these biotherapeutics tend to be more prone to in vivo degradation due to their highly engineered format. And therefore, there is a growing consensus on the need to characterize those biotransformations that are happening in vivo because they can induce structural modification of the biotherapeutic, leading to proteolytic cleavage or PTMs that might impact the efficacy or the safety of the drug. What we are currently doing to assess the in vivo stability is that we first perform targeted bottom-up, where we will select a set of reporter peptide that is scattered across the biotherapeutic construct. And then we will plot the concentration of those peptides over the time after administration. For a stable construct, those curves should be aligned. When it's not the case, as you can see here, we highly suspect in vivo instability and creating a need to analyze those samples at the intact level. But this comes with additional challenges. Indeed, we are working with extremely low concentration in the systemic circulation and the limited sampling volume, as well as poor fragmentation efficiency for proteins that are over 50 kilodalton. That's why we need to push the boundaries of sensitivity and performance in top-down MS. And in order to do that, we use the new Tim's Omni instrument from Bruker, which presents several features such as the Tim cell for ion mobility or the Omni trap to perform the trap EXD experiments, for instance. And we were especially interested in the OmniTrap platform. And if we take a closer look to it, you can see that it is composed of a series of segmented linear ion trap, which will allow us to perform different types of experiments. For instance, in the Q2, we can perform a resonant excitation CID. And in the Q5, we will be able to perform all types of EXD experiments, such as ECD or EID. And then in the Q8, we will be able to accumulate our ions for a really long time. The ions are able to travel back and forth in the OmniTrap, enabling large possibilities of combination for the fragmentation method within the same MSMS experiment. So before we moved to our precious in vivo sample, we first decided to optimize our fragmentation parameters on a commercially available mAb, which is the adalimumab. And what we have done is that we performed two types of digestion, which provided us an array of size of subunits in order to mimic what could happen in vivo. And with the same idea, we decided not to reduce the disulfide bond, which will inevitably impact the fragmentation efficiency. We evaluated 4 types of fragmentation methods, so ECD, ECD plus supplemental activation, EID, and resonance CID. As you can see, ECD plus supplemental activation provided the best sequence coverage. And once we combined all of the results from those techniques, we were able to obtain 73% sequence coverage for the 25 kilodalton and 37% for the 100 kilodalton. And we can clearly see that combining those fragmentation methods significantly improve the sequence coverage, and this is especially true for higher mass subunits. If we look at the combination of the fragmentation map of adalimumab, as I said, we did not reduce the disulfide bond, but this impacts the sequence coverage. But if we look outside of the disulfide bond, we were able to obtain 98% sequence coverage for the 25 kilodalton, so almost 100%, and also quite high sequence coverage of 83% for the 100 kilodalton subunits. And more importantly, for antibodies, we were able to completely characterize both CDR3 from the light chain and the heavy chain. So now that we were satisfied with our development, we decided to move to our PK samples. So we collected a mouse plasma sample at 4 different time points, so from 5 minutes to 72 hours after the administration of our tri-specific of interest. We then performed immunoenrichment using magnetic beads in order to fish out the biotransformation products. And from those immunoenriched samples, we first performed a profiling of the metabolites over time using an LC-MS system, which is the nanoELIT2 and the TIMS Omni, and all the data were processed with gene data. Then from this profiling, we selected metabolites that we wanted to further characterize in MSMS. And for this, we used the sample stream, which is an online desalting platform coupled to the TIMS Omni, where we performed an array of fragmentation methods. And we obtained a fragmentation map using data analysis and Omniscape. In order to have a reference on our intact tri-specific, which is highly asymmetric, as you can see here, we have 2 light chains and 2 heavy chains. We performed also the fragmentation methods, and we obtained 28% sequence coverage over all of the tri-specific. And we obtained the complete characterization of all 4 CDR3 for all the fragmentation methods. Then we moved on to the profiling of the metabolites. So here you can see the overlay of the intact mass measurement for the different PK time points, so from 5 minutes to 72 hours. And you can clearly see here at 5 minutes that the intact tri-specific is still quite present and intense, but it rapidly decreases at 4 hours. And then we start to see the appearance of primary metabolites, as we call them, where we hypothesize from the intact mass that it will have the structure of the loss of the light chain 1, which is the purple light chain. And then we will have the addition of a cysteine or a glutathione on the free tile from the heavy chain 1. And then at 24 hours, we start to see the emergence of secondary metabolites, which will have the following structure. We will lose the purple light chain, and we will have successive N-terminal cleavages in the heavy chain 1. We were able to detect 12 low abundance biotransformation products for this PK study, and the theoretical concentration of those metabolites will be between 3 to 30 nanomolar, which is extremely low. And we were able to hypothesize structure for all of the metabolites from intact mass measurement, but we wanted to further validate those structure using MSMS. Here I will only show the example for the metabolite number 8. So this metabolite is around 100 kilodalton, and we performed the 4 types of fragmentation technique. We were able to obtain 18% sequence coverage on the light chain 2 and 20% sequence coverage on the heavy chain 2. But more importantly, we obtained a really good characterization of the CDR3 from the light chain 2 and the heavy chain 2, confirming their presence. But we were not able to obtain any fragment from the CDR3 for the light chain 1 or the heavy chain 1, further confirming the hypothesized structure of the metabolite 8. And therefore, meaning that we have lost a two-target binding activity from the biotherapeutic. If I briefly summarize what I've talked about so far, so top-down MS approaches offered a non-parallel level of characterization for protein analysis at the proteoform level, bringing new insights into biotransformation processes. The team's Omni gives us an array of fragmentation method and therefore a really good depth for top-down mass spectrometry. And we were able to confirm the structure of our metabolite using multimodal fragmentation and show the evidence for the rapid loss of two target binding domain affecting the drug efficacy. And that will help us to further improve the structure of a biotherapeutic to optimize its in vivo stability. I would like to thank my two teams from Sanofi and from l'Institut Pasteur, and especially Alain Cric and Julia Chamourouk, my PhD advisor, the people from Fasmatech and Brucker who allowed us to use their instrument in Athens, and the people from Gene Data who helped me with the data processing, and also thank you all for your attention.
Great. Thank you very much. Really interesting data and good description of the instrumentation and its capabilities. Appreciated that. Does anyone have any questions for Lucile? I'm going to ask you an unrelated question. You said you're a PhD student, so what's next?
I still have 1 year left, but hopefully after this, I will keep working in the pharmaceutical industry and with biotherapeutics, and especially high resolution mass spectrometry.
Awesome.
Yeah, thanks.
All right. Without any further ado, we'll keep marching forward, and I will pass the mic to Corentin, who will talk about proteoform microheterogeneity assessment of antibody-based biopharmaceuticals using native top-down mass spectrometry. And I guess for you, it's good afternoon. So the floor is yours. You have until 11:50. I can see your slides, but not hear you yet.
Yeah, can you hear me now?
Perfect.
Okay, great. Thank you for the introduction. So yeah, as you mentioned, it's afternoon now. I'm a characterization scientist in the National Institute for Bioprocessing Research and Training in Nijmegen in Ireland. So today I will talk about assessing the proteoform heterogeneity in antibody-based biopharmaceuticals using a native top-down mass spectrometry. So as we all know, the manufacturing of monoclonal antibodies has matured significantly over the past decade, and many of the analytical workflow we use for the map characterization are now also well established and becoming increasingly routine. So this has been mostly driven by the advances in the analytical instrumentation, the separation chemistries, and the software that accelerated its move towards a more streamlined characterization. But despite those progress, we still rely on multiple layers and levels of characterization, from the intact mass down to the peptide mapping and the subunit analysis to fully capture the molecular heterogeneity of the antibody-based therapeutics. So this raises an important question. So as the molecules are becoming more complex, can our workflows become simpler? And specifically, can the high resolution MS and MSMS at the intact level give us a more direct view of the proteoform microheterogeneity. So native intact MS, especially when it's combined with upfront separation, is a very powerful approach for the complex biopharmaceuticals formats characterization. It's able to give many information, such as assessing the correct jet pairing in multispecifics, for example, preventing the loss of label functionalities in the conjugates as ADCs, and also able to identify specific proteoforms in the products. Of course, there is still some challenges that remains, especially for the highly glycosylated molecules and the other complex formats, where the heterogeneity make the spectral complexion, which can make the deconvolution kind of complex and difficult, or even impossible sometimes. So, so to try to perform native top-down approaches, we need to use an instrument setup that is really made for this. And what we need is an efficient transmission of the high MOVZ native ions, as well as the ability to isolate those proteoforms to then submit them to fragmentation and subsequent gas fractionation to simplify the spectrum. These capabilities together will enable the native intact mass to move beyond the simple characterization and confirmation of the mass to support the real proteoform level characterization of those complex molecules. So for this work, I was working on the arbitrary and biopharma tribute mass spectrometer, which includes a lot of activation techniques that you surely all know. But two key features here that were essential for this work were the quadruple capabilities to isolate high MOVZ ions up to 8,000 MOVZ, and also the high field arbitrary mass analyzer, which allows the identification of the ions up to 16 MOVZ. So as a preliminary work, we performed direct infusion analysis of trastuzumab in native conditions, and we can see here a beautiful native mass spectrum that we can get. And then perform the isolation of a single charge state using 100 Thomson particle isolation, as you can see on the screen. So with this, we were able to see the different glycoform on this molecule. After that, we performed the fragmentation of this charge state with the example of the HCD being displayed here on your screen. So as you can observe, we have a beautiful fragmentation spectrum, but some precursor signal and charge-reduced species are still present in the high MOVZ region. So what we can do to remove those unwanted species is to reisolate the fragment of interest using a pseudo-MS3 approach, which allows us to get rid of the precursor signal and also increase the signal-to-noise ratio and the spectral quality. So for the previous example, we were focusing on a single charge state, but multiple glycoforms. But what we can also do is to select a single glycoform using a narrower isolation window. So here we use the 5 Thomson isolation window and perform exactly the same type of experiments. So we can see that we are able to get the same good fragmentation spectrum of this species, and with the pseudo-MS3 approach, we improve the signal quality and get rid of the remaining precursor signal. So we perform the fragmentation of this single trastuzumab glycoform using various activation techniques such as ETD, HED, and UVPD fragmentation. And after the processing of the data, we were able to obtain a sequence coverage around 18% for the light chain and 15% for the heavy chain. So those sequence coverage are quite low, but here what we need to consider, and as Lucy was mentioning on the previous talk, that we still have the disulfide bonds that prevent the identification of the fragments that are inside these disulfide bridges. So what we can, when we look at those regions that are outside of the disulfide bridges, we can see that we have a good fragmentation and we have good coverage, as we can see here for the light chain and also here for the heavy chain. Additionally, we are able to get some fragment ions here, for example, that enables us to confirm the glycoform that we are seeing on the heavy chain. So with these results, we demonstrate that we can perform the native top-down on the intact knob and get insights on modifications such as the glycans, for example. However, this work was performed in direct infusion, so it makes the identification of proteoforms that bears modification of only a few daltons, for example, impossible to identify. So to kind of tackle this challenge, we decided to alternate the native MS with the pH gradient ion exchange chromatography and perform charge band analysis to identify the acidic and the basic variants, as well as looking at the glycoform distribution. So we used 5 different trastuzumab samples, the originator, and 4 biosimilars. And across the 5 samples, we see distinct glycosylation profiles. For example, you can see that for biosimilar 4 on the top, there is different glycosylation pattern. We are also able to see different levels of basic and acidic species, such as the diamidation of the pure glue modification, for example. So this first MS approach gives us a short variant comparability overview of the different biosimilars. Following this, we perform the native top-down, so the fragmentation of all these intact samples on all the chromatographic peak using ETD, HCD, and UVPD. So on the screen, you can see the average sequence coverage for all the samples for each fragmentation. And we can see that when we combine the different fragmentation techniques, we are achieving a sequence coverage around 30% for the light chain and 20% for the heavy chains. Also, if we look at the error bars, you can see a good comparability between the different samples, the sequence coverage being kind of the same. When we look at the fragmentation maps obtained for here is the originator samples, for example, we can really see a good sequence coverage of the regions that are outside of the disulfide bridges for both the light chain and the heavy chain here. As mentioned earlier, also, we are especially able to fully sequence the CDR3 regions, which are highlighted in green here and here for the heavy chain. And as we were observing for the direct infusion, we can also, on this main peak, fragmentation of the main chromatographic peak, see fragment ions that are confirming the glycosylation here. So in addition to the main chromatographic peak, we also perform the fragmentation of the acidic and basic variants. So here's the example of the diamidation of the acidic peak. On the upper panel, you get the deconvolution results of the main peak, while at the bottom in blue, you get the deconvolution results of the acidic peak. Those patterns are both for the light chain, and we can see that for the same fragment and the same charge state, so the fragment C99 on the left and B112 on the right, we have a slight shift of the isotopic profile here that demonstrates the presence of the diamidation on the light chain in the acidic peak. So thanks to the combination of these deconvolution results, the isotopic distributions, and the difference in the retention time that we get from the charge variant analysis, we are able to confidently identify the modification on the LC timescale. Another modification we can look at is the presence of the pure glue modification on the heavy chain. So after the deconvolution of the fragmentation spectra from the basic peak of the charge variant analysis, we can observe more than 30 fragments here that are confirming the presence of the pure glue on the heavy chain. Another interesting thing to notice here is that we see both the modified and the unmodified heavy chain fragments. So the orange one is the one of the heavy chain fragment ion B115 for the heavy chain with the pure glue, and the green one is the one without the pure glue. We can see those two fragments because we have 2 heavy chains, but only one of them is bearing the pure glue modification. So that's why we're seeing the 2 different isotopic profiles here. So with those 2 examples on the pure glue and diamidation, they show the ability that we have to characterize the proteoforms by native top-down on the chromatographic timescale. Then another case study I would like to show you is the characterization of a bispecific antibody, which is emicizumab, which is having two different heavy chains and two identical light chains. So when we perform the charge balance analysis, we can see a main peak here corresponding to the emicizumab with both chains having pure glue modification. If we zoom on the low abundant acidic species, we can detect low abundant afucosylated species first, and then also peak corresponding to emicizumab with a single pure glue modification. So we decided to have a look at these species and try to apply our native top-down workflow on this peak with only one pure glue modification. So for that, we applied different fragmentation techniques on this chromatographic peak between 9 and 10 minutes. So here you can see, for example, a fragmentation spectrum using HCD. Then we process the data we obtained from HCD, ETD, and UVPD. And we looked at the fragments that were identified for both heavy chains without the pure glue modification. We can see for the heavy chain A that there is several fragment ions here that confirm the absence of the pure glue on the heavy chain A. However, if we look on the heavy chain B, we can see that no fragment ions here were identified. Then we added the pure glue modification on the heavy chain B, and at that time we were able to see a lot of fragment ions that are showing the presence of the pure glue on the end term of the heavy chain B. So with these results, we show that we are it's more likely to happen that the absence of the pure glue is on the heavy chain A than on the heavy chain B. So to briefly summarize this presentation, we saw that thanks to new instrumentation, we can develop innovative characterization solutions for complex molecules. So we saw the proof of concept that native top-down MS can be applied to MAbs in direct infusion, and then combining this to charge balance analysis can be used for the proteoform identification and characterization at the intact level. We can also apply this approach to complex MAbs formats, such as the bispecifics, for example, which can be really useful to get more insight about their characterization. There are still some challenges for using these approaches. This includes the size of the molecule, their higher order structure, also that can sometimes prevent a good fragmentation and to get some information on the sequence. And with that, I would like to thank you for your attention. Also, all the people that took part on this work in Nybert and the collaborators in Thermo Fisher for the instrument setup.
Fantastic, great presentation and really interesting data. Looking at some of the PTMs that we all think about. If there are any questions, you can raise your hand or put them in the chat. I'm gonna. I wonder if you can. I wonder if you've thought about and can give me any thoughts on how you might envision this sort of data in a regulatory package, in a dossier to regulatory authority, and how they might react to that information, as it's relatively technical and sort of a newer approach than many regulatory authorities around the world think about.
Yeah, that's a really good question. To be honest, I don't think it's really ready to go for like regulatory agencies. They will not be really up to use this as it is. It's more like for early stage discovery and really research part of the discovery. But it's also really interesting, I think, as it's kind of allowed to link both the charge variants and of their characterization directly, while we're losing a lot of information when you're doing the peptide mapping. For example, you can't really link the information you get from the peptide mapping to the intact structure of the map. So that's the good point, I think, of using this approach and trying to develop this.
Fantastic. One technical question from the chat. What do UVPD30 or UVPD35 refer to?
It's the activation time, so 30 and 35 milliseconds.
Perfect. Thank you very much. So right on time. So I'm gonna do my best to keep us on time. So we're gonna keep moving forward, and we're gonna hear from Juan next on advancements in native mass spectrometry for ADC characterization. Juan, the floor is yours, and you have until 12:05 my time, in Eastern time. Excellent. Let's see your slides. Cannot hear you yet.
You can hear me now? Got me?
We got you.
Okay, sounds good. All right, so thanks again to Paul and the organizers for the opportunity to present some of the work that I've been doing here. I'm a scientist at Pfizer in research and development. And so getting into introduction to native mass spec. So as we're aware, we measure the mass to charge of biomolecules in their native folded state. This is really important for the characterization of antibody-drug conjugates, specifically, as we had heard from Jack in the first talk today. Certain antibody-drug conjugates utilize the native cysteines that normally covalently hold the mAb backbone together through interchain disulfide bonds. And so if we're using those cysteines to couple cytotoxic drugs onto them, then the entire antibody-drug conjugate complex is held together through non-covalent interactions. So then native mass spec becomes really important for being able to measure and obtain intact mass information on those molecules. And so we're able to get information using native mass spec on protein complex assembly, molecular mass, stoichiometry. For ADCs, a critical quality attribute that we're very concerned with measuring and keeping track of is the drug to antibody ratio, or DAR, which essentially refers to how many drugs are on a particular antibody backbone. And so what we can see, let me grab my laser pointer here. Down here is, I'm showing two sets of data. One is HIC, which is hydrophobic interaction chromatography, and that's typically what's going to be a release assay for us in the pharmaceutical industry when we're presenting DAR data. We use HIC as the release assay to show that we can measure the DAR and have characterization of those values. And ideally, we would have excellent correlation between native mass spec, which is shown on the right side, and the DAR, and those values are going to agree in terms of abundance of the various DAR species and the average DAR. And so this data is coming from an average DAR4 antibody-drug conjugate. But as you know, so ideal case, everything lines up nicely between HIC and native MS, but as we move to more complex modalities, say for instance, we put a charged drug linker on there, that can really impact the performance of HIC to obtain DAR information. So then everything might just dilute out in one peak and we sort of lose that valuable DAR isoform information. But native mass spec is still able to provide masses and abundances for all the various DAR species. So that just illustrates the power of this technique and an application where it becomes extremely useful to have. So a bit of overview of the method that's typically run, and it's fairly simple. So we would take a mAb or ADC, concentration range, around 2 mg per mil or so. Typically, we're going to remove the glycans before native mass spec, so we'll use PNGSF to clip those off. And then we will separate them using native size exclusion chromatography in an ammonium acetate buffer. Typically, those concentration of that buffer is somewhere between 50 to 200 millimolar. and will flow anywhere from 100 to 200 microliters a minute. And basically everything's going to come out in a single peak. And we also collect the UV data as well as get the total ion chromatograph data from the MS. So if we zoom in here, under every chromatographic time point, we get this distribution of different charge states for the ESI. So typically, we've definitely relied traditionally on QTOF instruments to do these native measurements. So I'm showing a couple examples of QTOF instruments that we have in our lab here, the Agilent 6545 and the Bruker Maxis QTOF. Those have definitely been workhorses for doing these native MS measurements on ADCs for many, many years. And we can see here the listed resolutions of those instruments. And then on the right side, I'm showing newer acquired hybrid Orbitrap instruments like the ThermoSend Biopharma and the Thermo-Oxidian. And those, as listed, have much higher resolving powers or resolutions. Although I think, as all of us know in practice, working with mass spec, these resolutions tend to be more of a marketing tool than anything from the instrument vendors. But they do also have a real impact on the data that we see. And so I've listed here the equations that govern the resolving power for TOF instruments versus Orbitrap. So we see for TOF, the resolution is effectively scales with the length of the flight tube versus Orbitraps where the resolution is proportional to the inverse square root of the mass to charge. And so I'm showing this chart here from this reference where we can see that one reason why TOFs have been the traditional instrument of choice for that is because as you go out to very, very high MZ, because the length of the flight tube is fixed, you should have basically a fixed resolution. But we know in practice that's not, that's the theory, but in practice there are factors that actually decrease that. And so I'm showing some examples here for the same antibody measured on different instruments. The one on the left and center from TOF instruments, and then on the right I'm showing an example of that same molecule measured on an Orbitrap. And I think, based on these small images, you can already appreciate the increase in resolution that's coming from these newer Orbitrap instruments over the TOFs. So we've already seen the architecture of the Thermo Orbitrap Ascend, but here I'm showing it again in the schematic form. And there are several aspects to this instrument that really end up in improved native mass spec measurements. So we have a soft ionization electrospray source here with this funnel that guides the ions through. Then we have the quadrupole mass analyzer that's able to filter out ions and do selection. And here we have a range of ion routing multipoles, and then we have this ultra high filled Orbitrap mass analyzer where the high resolution mass measurements are able to be made. We also have a linear ion trap back here, which can, as we saw in the previous presentation, be used for isolation and fragmentation for doing top-down measurements. So having this hybrid mass spectrometer with 3 different mass analyzers really provides a wide range of options for doing some really cool mass spec measurements on these molecules. So here I'm going to just show some data as sort of a reference standard for assessing the performance of the Orbitrap using just a standard MAB. And so this is a glycosylated MAB, and we can see the various glycoforms. So the most abundant glycoform here is the G0F, G0F. And then we have, we can pick out peaks for all the various different glycoforms, including the G0F agly minus glycnac. And we can see the theoretical masses and the observed masses and how tight the mass accuracy is here. So all of these peaks are being measured with really, really nice mass accuracy, less than 20 ppm for that. So this is just assessing the performance of the instrument, and we can see that definitely it's passing all of our criteria for being able to measure these glycoforms on the MAB with high mass accuracy. So then getting into the ADCs, Here I'm showing a direct comparison from the QTOF, which has been the traditional workhorse, and the Ascend. And it's just a stark contrast in terms of resolution that we get on these individual DAR species. So in blue, we see the native mass spec, deconvolved mass spectrum from a DAR4 ADC. And then on the bottom, we can see that same molecule, the data collected off of the Ascend. And zooming in on the DAR4 here, you can really see the improved resolution and sensitivity that we're getting off of the Ascend. And so we have the primary DAR4, and then this peak here is actually the glycation peak. So it's a 162 Dalton shift off of the main there. And we can get basically baseline resolution. So then, you know, what this allows is us to get much higher resolution and characterization of various PTMs that could exist on any of these DAR species. So looking at the mass accuracy for how it looks for the DAR species on a deglycosylated ADC. So here's that distribution where we have 0 drugs, 2, 4, 6, and 8. And we can see that all of those have been measured with very high mass accuracy, again, well under 20 ppm there. We also can see that we get some loss of the drug. So these ones that I've labeled as like DAR3 WHL, that refers to warhead loss, where we're getting some in-source fragmentation and loss of the part of the drug linker there, but those are quite small. So we can't completely eliminate that for some drug linkers that are particularly labile during the ionization and transport, but this is looking by and large very good. And so, so mass accuracy is looking great. We wanted to assess the robustness of this Ascend platform for doing DAR measurements over just many, many injections. And so the plot on the left that I'm showing here is, you know, over 40 injections, just looking at the overall area that we're getting, we can see it's stable. And then again, comparing back to HIC and native, what we want to see is a good DAR distribution, good agreement in terms of the intensity of the various DAR species, but with HIC and native there. And so we can see that they align very nicely. So one thing about the Ascend is that it definitely provides a softer ionization. So we're getting overall less in-source fragmentation. So this top spectrum, you can see if you scan down at lower MZ, you get this distribution here that's circled in red. And what that actually ends up being when you deconvolute is light chain that's falling off of the molecule. And so we like to minimize that as much as possible. And we can see that on the Ascend platform, we get much less released light chain. So that's good. That improves our overall sensitivity and signal of the intact ADC, which we're most interested in. So another thing that we wanted to screen on this instrument was the in-source source fragmentation energies and look at the impact that that can have on DAR. And so here I'm showing 2 plots for 2 different ADCs. And what we can see is that on the x-axis, as we increase the source fragmentation energy, it actually increases the DAR to a certain value, and then it kind of levels off. So during this optimization for both of these ADCs, we settled on a source fragmentation energy of 140. That gave us the most accurate DAR that compares back to our HIC values. So having this improved resolution, as I said in the beginning, typically we deglycosylate the ADCs to simplify the measurement, but then you know, with the TOF instruments, using measuring glycosylated ADCs was kind of out of the question. We just didn't have the resolution, and so all your peaks are going to look like big blobs, basically. But having the improved resolution of these new Orbitrap platforms, we asked the question, can we do these native measurements on glycosylated ADCs? And the answer to that is yes, we can. And so here I'm showing you DAR4 native mass spec of a glycosylated ADC. And so we can see the various DAR species, and then overlaid within each of the various DAR species, we can see the various glycoforms as we saw for the MAB species. So we can see, again, the For the DAR4, I have them labeled here. So the primary one is the G0F, G0F, and then we have G0F, G1F, and so on and so on. So we can zoom into that a little more and see a lot of detail there. So really impressive sensitivity and mass accuracy, and we're able to pick out a lot of minor glycoforms. We can get the A glyforms down here. We can see some warhead loss of GLICNAC. And again, all of these peaks, by and large, measured with really nice mass accuracy, all the way down to these very low abundant glycosylated DAR isoforms. So one question we might ask is, how do the glycan levels compare between the parent mAb and the ADC? So prior to conjugation and then after we've conjugated it, and how do those compare? So this is kind of getting at a question of, could there be interactions between different glycoforms and the drug? And do we get any enrichment there? And what we can see is by measuring the native mAb and then the conjugated ADC, we can see that in dark blue, we have the abundances coming off of the mAb, and then the lighter blue is showing the ADC. And by and large, very nice agreement there between the mAb and ADC. So in this particular case, we're not really seeing an impact on the glycoforms with on the drug conjugation. So in summary, we've now shifted to using native SCCMS on thermo Orbitrap instruments, and that's providing us accurate DAR for ADCs. We're getting much higher resolution and signal to noise than we were previously getting off of the QTOF instruments. We've seen that these new platforms are robust over many injections. We're able to do cool things like measure glycosylated ADCs, and going forward, the Orbitraps will be the new standard platform for doing characterization of ADCs in our lab. So on that note, I'd like to thank the members of the MS core here at Pfizer, leadership in CASBOT, and then my colleagues in Andover as well.
Fantastic. Thanks, Juan. We're a few minutes over, so we'll hold the questions for the panel discussion at the end. I'm monitoring the chat also, and we'll note those. We will keep moving forward with Shannon now, who will talk to us about native MS for AAV therapeutics, for insights and future opportunities. Shannon, you have until 12:21.
Thanks for the extra minute. Okay, thank you everyone for being here, and thanks to the consortium for inviting me to talk to you today about some of the work that we've been doing with AAV therapeutics and development. So my name is Shannon Rabb. I'm from Eli Lilly in Indianapolis, Indiana, and I've been at Lilly for about 5 years now and been doing mass spec for about 10. So I love this space and definitely excited about all the presenters and the discussion later. So first, I want to give you a little bit of insight, if my slides would go, about where I sit at Lilly. So these are our different departments in the company. So we have discovery, and then we have a new genetic medicines institute that's rapidly growing. Of course, we have toxicology and devices, and I sit over here in process and product design, specifically in our bioproduct research and development organization. So basically, if it's grown inside of a cell, then that's what we're going to develop in our department. So if you think of kind of the linear development of a drug, it starts in discovery, goes into lead optimization and preclinical studies, and then product development, which is where I am. We take it from clone selection, and then we do concurrent, development while the clinical trials are going on, and then we take it all the way through product registration. So I really enjoy where I am. I get to work with a lot of different groups. So the device group, formulations, of course, our cellular biologists and purification folks. So specifically, I sit in the protein characterization team. So we are primarily a mass spec focused group. So we provide molecular level structural insights to our molecules that we're developing, and we handle anything from gene therapies, which I'll talk about today, to small peptides also, grown in cells. So since we sit so far through development, we provide structural characterization packages for successful regulatory approvals. So the investigatory new drug applications, and then eventually the biologic license application, BLA. And then another core thing that we do is develop new technologies. So we get to have a lot of fun with our 20 to 25 different mass specs in the group, and also collaborate with academia and our regulatory colleagues too, to bring mass spec more into the space. Okay, so today I'm going to be talking about AAVs. So AAVs do represent a major modality for gene therapies. And AAVs have this capsid structure that is made up of different proteins. And inside of the capsid, you have your genome. So your capsid is responsible for trafficking your drug to the correct location, and then it releases the genome, which can then do gene editing inside of your cells. So the first FDA-approved AAV therapeutic was in 2012. And then, as you can see, as we've gone throughout the years, there's been a lot of development with these molecules. And now we have over 9 or 10 AAVs that are FDA approved. And these are often, diseases and issues that people previously to these medications didn't have any therapies that could target them. So things like spinal muscular atrophy and retinal dystrophies. So these are things that we're starting to target with AAVs. A little bit more about the structure. We are a mass spec group. We're very interested in the structure of these things. So here's the overall structure. I mentioned you have viral proteins. So you have VP1, 2, and 3. And they're all, they have a conserved seed terminal domain, and they all vary in their N-terminal domains via truncation of the VP1. And these assemble in a 5:5:50 VP1, 2, 3 ratio to form the capsid. So the different VP sequences determine the tropism, so where it's going to traffic inside of the body. And then these unique regions of VP1 and 2 play crucial roles in things like endosomal trafficking, nuclear entry, and also endosomal escape. So it's really important to understand the conformation of these things. And then I wanted to highlight some analytical challenges, mass spec focus. So AAVs are very large. So when the genome is inside, these capsids can be upwards of 5 megadaltons. So very large and also very heterogeneous. So you have critical quality attributes such as the empty partial full, that VP ratio that I mentioned can actually vary quite a bit, and then different post-translational modifications as well. So measurement of these different CQAs of these large particles is very difficult in the GMP space. And especially these are also a very new modality for the FDA. So the regulatory guidance around these modalities is still evolving. So I think there's also, challenges around global harmonization, around standard sample processing, data processing, and controls there. I think, is kind of a challenge. And then, of course, MS tools for AAV characterization are also new and require additional testing, and I'll talk about that a little bit. But these are the different critical quality attributes that we're starting to monitor with these therapeutics. And in our group, we're very interested in utilizing mass spec as a multi-attribute method, where we can start to replace some of these traditional analytical methods with more mass spec methods that can give you multiple attribute information in just one assay. So here I've bolded some of the attributes that are related to like native mass spec and keeping the structure integrity of the molecule. So empty partial full, typically we'll look at this with AUC, TEM, or AEX, and we're starting to look at CDMS for these measurements, and I'll show a lot of that data. And then there's some conformation considerations that I mentioned, and we can use FPOP or HDX to look at some of those attributes. And also the genome identity is becoming increasingly important and difficult to measure. So we like to look at that with CDMS and peptide mapping also. Okay, so I am a native mass spectrometrist. So when I first got my hands on an AAV molecule, I wanted to just spray it into a mass spec and see what I could see. So this is a very simple assay where I'm just taking some AAV, I'm doing an offline buffer exchange, and then using a static nanosource with our ultra high mass range UHMR thermo Orbitrap platform. And here I've shown two different standards. So these are from commercially available from ViraVec of AAV9. This is a certain serotype, AAV9 empty. So this is with no genome, and then AAV9 filled. So this is with one genome. But you can also see here we have some AAVs also will package two genomes if your genome is small enough to fit in there, is to have two in there. So I was very excited to see some separation between the empty and filled capsids, even on just an M/Z scale. And this is likely because the charge on your empty and filled capsids likely is very similar. So you're able to separate these relatively well in this regime just based on their mass. So this was very low sample consumption. As those of you who have done static nanospray before, you know it only takes like 10 to 20 microliters, and it's also very sensitive. However, this method lacks resolution, specificity, and robustness. You can tell like how broad our peaks are. You get no isotopic resolution and no information on any of the PTMs or anything like that. So in order to decouple the mass and the charge measurement, several new approaches have been introduced to the field in terms of a charge detection mass spec. So on the left, this is kind of the traditional CDMS developed by Martin Gerald at IU, and he now runs a company down there called Megadalton. So this is CDMS using an electrostatic linear ion trap. So here my animation isn't working, but this little AAV particle oscillates back and forth in this trap and imparts a charge on these end caps. So the frequency of that oscillation gives the M/Z, and then the magnitude on the end caps gives the charge. So you measure these two things directly, and you can multiply the two together to get a true mass spectrum. So Thermo has also come out with a direct mass technology, DMT, using the Orbitrap analyzer on the UHMR. So the approach here is to scan hundreds of individual ions in your Orbitrap, and you do this by either defocusing your ions or diluting your sample just to get a few ions in your trap. You create a calibration curve of known standards to see how your Orbitrap is responding to different charges on that you introduce it to. And then you assign the charge using the calibration curve for your samples. So first I'll show you a little bit of data from the ELIT technology. So here we have the same empty and filled samples, and now we're on a mass scale. So for the empty, we see a mass peak at 3.67 megadaltons, which fits very nicely with our theoretical. And then for the full as well, we see the one filled and then the two genomes filled, as I mentioned. But what's really nice about this is we can get a charge versus mass plot. So now we can see the individual charges for each of these ions. And now we see a population up here that's indicative of a dimer species. Similarly, for the filled, we can see the empty, partial, and filled species. So this dimer population is really important to us. So we don't have a whole lot of other techniques to be able to see this type of species. And I want to highlight a paper from 2021 from Lauren Barnes that showed a different conformation of a dimer. So here at this mass, we see the dimer, and then at the same mass but a higher charge, we see another population. So this is a dimer with a different conformation. As you change the structure of your capsid, it can pick up additional charge and result in that different conformation that you see in the CDMS. So this is great, and actually Waters has just commercialized this technology, so we're really excited about that. But one of the challenges that I still think has yet to be fully addressed is getting the samples into the mass spectrometer in a native manner and in a very robust and quick manner. So because of this, we wanted to look at the sample stream. So we worked with Neil Kelleher's group, Dura Kafader and Sam Janice, to look at these AAV molecules with their sample stream platform. which I believe Lucile talked about earlier as well. So here we're just taking our AAVs, directly infusing them into the sample stream, and it does a concentration and buffer exchange, and then we put them into the direct mass technology. So here I was really excited to see these traces. This is the blue is just the nanospray with an offline buffer exchange. The green is sample stream with offline with no offline buffer exchange, and then the purple is offline buffer exchange with the sample stream. So you see the traces are very well aligned on top of one another, which is very nice to see. And then they also tried some different ratios of empty partial full and found that it was very accurate when they accounted for the different titers. So finally, we wanted to see if this platform could look at AEVs in different buffers. So we sent them some Lilly sample, and it looks very good. It aligns very nicely to our other orthogonal methods. And then they tried some AEV in media, and these different traces are just different wash buffers on the sample stream. And you can see that in the media, your trace with the lower amount of wash buffer is kind of difficult to see, although you still do get pretty good quantitation of your empty partial full. But you can increase the number of washes that you do, and you see you get pretty much the same trace that was obtained from ammonium acetate in the cell media. So this is really exciting, and we can start using this for process development. So just quickly, I've shown you that AAVs have diverse quality attributes that lend a lot of opportunity for native MS development. So mass conformation and structural analysis is obtainable through CDMS. And I think that native MS interfaces such as the sample stream can help provide really reproducible and robust automation for these. But I did want to lend some opportunities for the future. So I do think that it's still a challenge for lipid nanoparticles, RNA and DNA, and other extra large molecules to have robust mass conformation. So these are the new modalities that we're looking at now. And of course, standardized native mass spec workflows are essential for their use in process development, and especially GMP implementation across the industry. I've highlighted the need for hyphenated native mass specs and even CDMS techniques. I think it's important to have those for comprehensive characterization. So I enjoyed some of the talks that showed that today. And then with the advent of AI, of course, better predictive tools and data processing software is always a plus. So thank you very much. I hope you enjoyed.
Thanks, Shannon. If there's questions, you can put them in the chat or hold them. We'll do that at the panel discussion. I will tell you, Shannon, you said lots of trigger words to me that are in my own life. I was at a conference in China last week with one of your colleagues promoting carbonization in the Asia Pacific region for these types of approaches. So it's a great presentation, and look forward to chatting more in the panels. But for now, we'll move forward and go to Christopher, who's going to tell us about. Nope, I lied. We're going to go to Bifan, who's going to tell us about toward comprehensive characterization of guide RNA and beyond by top-down mass spectrometry. I got your slides, and I can hear you perfect, so you're good.
Perfect. All right. Thank you so much for the introduction and the invitation for the opportunity to speak here today. My name is Bifan. I'm from Genentech. I'm currently working in the synthetic molecule analytical chemistry department under the big umbrella of pharmaceutical development. We are a small molecule group, mainly supporting the delivery of high quality clinical material to clinic, responsible for the chemistry, manufacturing, and control. So today you will continue to hear about the new modality regarding the guide RNA, which I will touch base in a little bit. Before I go into the actual content, I would like to start off by acknowledging some people. You can see Chris Crittendon's picture here is my partner in crime. We did a lot of work together, and including this top-down work on the oligonucleotide. And also our collaborator from Thermo, especially Josh, who we work together on alternative and new fragmentation technology. So why do we care about single guide RNA? because the guRNA is one of the critical reagent that governs where the CRISPR enzyme that can cut in your gene of interest and potentially swap it with new genetic information. This can be used in T cell therapy, for example. You can knock out the endogenous T cell receptor, and with a DNA plasmid incorporating the desired gene information, you can design a new neoantigen recognizing a T cell receptor. And the guRNA here plays an important role, as the quality of the guRNA can affect the efficacy and accuracy of the cutting. The reason why the guRNA would end up in our synthetic small molecule shop is that it's usually made by solid state phosphoramidite chemistry, so synthetically made. And it's no longer traditional anything small, but a large size 100 nucleotide long biopolymer, about 32 kilodalton in size. And here's the structure of it. You see the normal nucleotide phosphate backbone, ribo, and then the base. And these synthetic oligonucleotide are usually chemically modified so that the stability can be improved, including the phosphorothioate and the 2'-O-methyl. Within our department, we have existing efforts to how to characterize these, and these are with regulatory agency expectation to confirm we are making what we are making in the sequence. Within the department, we started off using a more traditional bottom-up approach where multiple enzymes are used to digest the 100-mer, and you can map it sort of like peptide mapping back to the whole sequence. We later have a middle-down primer protection approach where we use specific primer to block part of the sequence so that the enzyme we use will not make as many cuts to make smaller pieces. This way, we sort of get away from the inferring problem where you have ambiguous assignment of the small pieces back to the 100-mer sequence. What Chris and I really strive for is to explore if we can directly fragment the whole 100-mer sequence in a top-down manner so that we don't have to deal with offline sample handling and digestion. Given the 30 kilodalton size, it's for top-down, it should be a comfortable range that we can do direct sequencing. We soon find out that the challenge for doing intact top-down on larger oligonucleotide is coming from the heterogeneity you can get from the metal adducts, from sodium or potassium. It's really prevalent from other glassware, your system, even your spray needle. It's very easily contaminated with all these metal adducts. And the result of this is that you get all sort of number of these adducts spreading the signal down, making the isolation and top-down analysis difficult. There are ways to get rid of it in the literature. You can use molecular cutoff filter, or you can use precipitation and swap it into the buffer of desire to analyze this offline usually. You can also alleviate the problem by doing periodic acidic wash from your typical pairing reverse phase, but the problem will come back if you don't continue doing that. One way we started thinking of doing this is, can we leverage how native mass spec using SCC to introduce the sample and get rid of other sol contaminants or adducts? The way we do this is, from literature, we know that 20 millimolar piperidine in organic solvent can swap out the metal adduct very well. However, it's in very high pH, and this will not be compatible with any commercial SCC column out there. The thing we ended up using is using a C18 hybrid, metal organic hybrid C18 column that can sustain high pH. Because it's a hydrophobic C18 and the oligonucleotide is so hydrophilic, essentially it doesn't have retention. And when the pore size is small enough, we can sort of use it as an SEC. This way, we swapped the piperidine away with all the metal adduct, and we ended up getting very nice desalting results from this easy, quick SEC workflow. And the sample introduced into the mass spec, we can do downstream top-down analyses. Here, just an insert showing all of a sudden now you are able to see the +16, +14 modification that's present on the oligonucleotide, which otherwise cannot be seen if you have all the other metal adducts. By doing this, we effectively increase the sensitivity by at least 4 to 5-fold, which is beneficial for any downstream analyses. Here, I want to show you a quick single-run sequence coverage by using CID, where we get 67% sequence coverage, accounting using all sort of fragment ions that you see from here. By combining, like many others do, with alternative fragmentation technique, we can improve the sequence coverage up to 80%, combining 3 runs from CID, UVPD, and AEPD. Something we soon realize is that having more fragmentation ion in fragmentation type in the case of oligonucleotide does not mean you always get better sequence coverage. This is because all these oligonucleotide in active mode are highly charged, as you saw from the previous slide, and the spacing for the fragment ion to fall into are relatively limited. Here's an example of a UVPD spectrum where we see these radical charge-reduced species, and you see a typical the grass hiding under all these big trees problem from top down. And because of the different type of fragment ion, this problem just become even bigger. Another thing for these oligonucleotides, here's a good example from Josh from Thermal, is that oligonucleotides are very easily fragmented by a traditional collision-based fragmentation approach. Here's an example. By using a 16 NCE, we are able to get a reasonable spectrum. Here's the precursor ion, and if you go a bit lower, you seem to underfragment. And if you go slightly higher, you don't see the precursor ion anymore. It seems to be overfragmenting. If we zoom in, you can see the overfragmentation, but among all 3, you see similar information. So we're getting similar information, and you can easily overfragment or underfragment in this case. Something worth working together with Thermo we've found out is that by attenuating the Q value in a CID type of experiment, it works way better than changing the NCE value, where you're just changing the amplitude, magnitude of the auxiliary voltage that you're applying. This one, by changing the Q value, you're changing the resonance frequency to a lower value. It's a milder fragmentation that it generates a lot more useful fragment ion. Here's the drastic comparison between these. Even for higher Q value CID, you are seeing overfragmentation. And these overfragmentation can be detrimental for oligonucleotide sequence mapping because you're only having 4 or 5 bases. The likelihood of getting some false positive, it's very high. Knowing this, we apply this to our 100 ml oligonucleotide, where we want to manipulate the solution, the mobile phase that it's in, so that we can move the precursor ion to a higher MOVZ value so that we have enough space for the fragment ion to fall into. One signature feature that we saw from this low Q CID approach is that we start from getting primarily base loss from the precursor ion, followed by very rich information of predominantly CY type fragment ion. So using this approach, we're not getting into the other fragmentation channel, forming other type of fragment ion, so that under this large MOVZ space, it's predominantly, it's only CY ions. And here's a zoom in showing you how well this works and how dense the data could be. You can really start doing de novo sequencing in some sense, just to walk across the sequence. And the overall sequence coverage using this approach on this 100 mer is extremely good. We're nearly getting 100% sequence coverage, except two bonds that were not cleaved in this case. And this is a bit forward-looking. The guide RNA are getting more and more complex. For example, in the case of base editing, you can get another portion of this getting together with the guide RNA sequence, doing the reverse transcription part of the gene editing. So we want to test if this could be done on even larger stuff. And we started off from a 150 mer RNA. The cleanup works just as good as the 100 mer. We tried on a 200 mer DNA. It works really well. Here's the sneak peek of fragmenting a 150 mer RNA. We're still predominantly getting a C-type and Y-type ion. And you see how dense the data is. And we think we can really push this forward if we can move the precursor ion to an even higher MOVC value, giving it more space. With that, I would like to conclude that hopefully I've shown you top-down MS with the low-Q approach really can provide high sequence coverage for a synthetic oligonucleotide up to 100 mer and beyond. And this is benefited from the really easy column-based cleanup approach that get rid of all the adducts. And by limiting the fragmentation channel using the low-Q CID approach, everything you will get improvement compared to simply adding more fragmentation approach and combining them all together. With that, I'd like to thank you for your attention, and I can take any questions later.
Yeah, sounds good. Thanks, Bifan. We're going to take questions later. So if you have them, go ahead and put them in the chat. But otherwise, we'll just keep moving forward. So we have plenty of time at the end for that conversation. So with that, next up is Christopher, who's going to talk to us about native mass spectrometry for mapping antibody aggregate species and comparing intra and extracellular profiles.
All right. Oops. Sorry, 1 second. I'm not sure. Trying to share my slides here.
We had your screen a second ago, but we don't know.
Okay, it's back.
It's coming. Yes, there you go. You're good.
Okay, sorry about that. Thank you for the introduction and the opportunity to speak here today. I'm Chris Sauer. I am from Johnson & Johnson Innovative Medicine, and I'm going to be talking to you today about how we use native mass spec, specifically looking at aggregate species. So I work in our cell engineering and analytical sciences group. This sits somewhere between discovery and early development. We're very closely tied to our cell line development colleagues. So our main role as mass spectrometrists is to provide analytical characterization of not only the biotherapeutic itself, but as well as all of the cell lines being developed, any media conditions, culture conditions, et cetera. So we're looking at everything from metabolites to peptides to intact mass and native complexes. And our primary objective is to work closely with our cell line development colleagues to create cell lines with high product quality, high titer, and optimal growth profiles. Native SEC has become one of our most critical assays for this, especially as Johnson & Johnson has been moving towards more multi-specific antibody targets or products. So with like traditional reverse phase methods, you'll end up with a singular chromatographic peak as everything kind of goes back to this standard asymmetric mab here. But with native, we're actually able to really tease apart what's going on in the cells and get a lot more information from that. We can see if there are homodimers being formed that might escape purification. We can see if there is a half mab or singular chain being formed. We can observe clipping. And then one thing that we're always interested in is the aggregates that form and the nature of those. So with our size exclusion run, this is, of course, the high molecular weight species to the left of the main peak and the low molecular weight species to the right, and then our main peak here. Using a native approach is something that is reasonably straightforward, but benefits extensively from your instrumentation. We use a thermo ultra high mass range, or UHMR. This is part of their Q-Exactive series. And it's been developed with large mass analytes in mind. So everything from the ESI source to the mass analyzer has been catered towards large mass ions. But still, optimization is absolutely critical. I've explained in previous talks of how that optimization benefits, but I'm just going to briefly summarize that here in the interest of time. So looking, say, at the very beginning, you have your source parameters, and really just taking the time to optimize your, even just simple, like your gas flow, your capillary temperature, and things like that is really going to improve your overall analytical capabilities. So we've injected the same sample multiple times, just varying our source parameters. And you can see this peak at 20 minutes is this is the tick for the dimer. And you can see as we increase our gas flow, we can improve our desolvation. And that can give us a really nice, clean signal for our dimer. If we increase the capillary temperature and the auxiliary gas temperature, even like 300 and 250, not overly high, but a little bit higher, that can help with desolvation. But at the same time, then we start to introduce in-source dissociation, and that can really start to break apart these native complexes and complicate your analyses. Conversely, if you go too low, you run into the opposite problem where you don't have enough desolvation and you get a rather messy signal. This is something that's going to depend on your analytes, it's going to depend on your buffers, et cetera, but really something that has a lot of impact on your analysis. Additionally, if we then go to the other end of the instrument, to the mass analyzer, one of the biggest parameters you can adjust is the resolving power. And as someone else earlier today was mentioning, resolving power is oftentimes something that is a bit nebulous. Sometimes it's oftentimes best to look at this from an experimental standpoint. So we were actually looking at how does our raw mass spec signal change with increasing resolution? What does that mean for your deconvoluted mass spec? And then your overall mass error. So one of the things with an Orbitrap mass spectrometer is as you increase your resolving power, you are increasing the amount of time spent in the Orbitrap, which is going to not only increase your scan time, but also lead to transient decay over time. So you're kind of playing a game between your mass accuracy and your overall signal. So what we can see here is as we increase our resolving power, we start to get more and more noise. And that leads to deconvolution of just a jumbled mess compared to at the very low resolving power, we're also kind of getting no distinguishing information. So there's sort of this optimal range here. I also show this as the black line up here. We have the base peak signal, and you can see that tends to decrease over time, again, from the transient decay. And your mass error also decreases as that resolving power increases. You'll get a tighter mass reading. But then as you start to increase your resolution too high, that signal to noise goes way down and your absolute mass error jumps back up. So there's kind of this green box here where you have that optimal range for most analytes of resolving power, of your balanced base peak signal and your mass error. So this overall optimization process led us to creating a dual acquisition method where we actually have throughout our chromatography, we have sort of a pattern of your monomer eluting at about this time, and then you have your high molecular weight species eluting before that, and we actually have the method switch in our method file. So your high molecular weight species are going to get collected at a lower resolution with a higher scan rate, greater gas flow to help with desolvating those larger complexes. And then your lower molecular weight species, including your monomer, are going to be collected with a higher resolution so you can get a little bit more information on things like glycoforms, potential clipping, etc. So you can see here that by doing this, we have our tetramer, trimer, dimer, all able to be confidently identified just from like that raw mass spec. You can see that overall signal is very high and interpretable with your tetramer, your trimer, your dimer. You're not going to get a ton of mass accuracy on this, but you will have a confident identification of those multimeric species. Then you go to your monomer, and because you're at that higher resolution, you're able to, again, see glycoforms. You're able to see confidently identify any low molecular weight impurities. You can see clipping, etc. So we wanted to apply this to a intracellular versus extracellular aggregation study. And what we were able to see is so what we did is we had our cells lysed under non-denaturing conditions, and then we looked at how do the cell lysates compared to the cell culture supernatants in terms of the aggregation profiles, and what does that mean over time, and how can that influence studies? So what we saw with the triangles here in purple, we were able to see, this is the supernatant data, and you can see the aggregation actually increases in those first few days, and then over time decreases. Likewise, in the cell lysates, we actually don't have enough cellular content. Those cells haven't grown and multiplied enough. That days 0, 1, 2, and 3 really don't have meaningful data. So we start at day 4, and you see that same pattern of increasing and then kind of maintaining and decreasing again at those later days. Additionally, again, because we have like the identification of those aggregates, you can see the superlysate information overlaid like this. But we also are able to, you can see how that monomer mass spec deconvolution is going to differ between the sup and the lysate. So we're able to, with that higher resolution scan on the monomer, we're actually able to identify the different glycoforms and how they change. And one thing that we are seeing in the lysate that we found very interesting is the presence of these high mannose species compared to the supernatants having the high G0F glycans. Zooming in on that, we can see how these two are overlaying, and you can see that you have you essentially have man 4 to man 9, and every combination of that in your cell lysate. But in your soups, you really just have the G0 through G2F, again showing the every combination of that. What's also interesting is with how high of a resolution we have, we're actually able to see this peak splitting where you can start to see that your G0, G0F, your G0F, G0F, etc. overlaps with a lot of these high mannose glycans, and you can start to see these are actually overlapping. So we do have both the G0F and the man 6, man 7 glycoforms on top of each other. So when we actually looked at this, we were able to see that in the endoplasmic reticulum, you are looking at this process here where you are going from your man 9 is when the glycan is first added as man 9, and then it goes all the way down to MAN5. And then in the Golgi apparatus is where that glycan is modified into its mature form, your more galactose species, your G0, G0F, G1F, and G2F. So what we were actually seeing in the lysate is a snapshot where we were getting a full distribution of this enzymatic process. We then looked at that under higher resolution using a reduced mass method. So we were actually looking at each chain on an individual basis. And that helped us to tease apart, say, G0F from MAN5, and those combinations were no longer overlapping as much. We saw that heavy chain 1 and heavy chain 2 showed very similar patterns where over time you kind of have that mature glycoform in the blue and white here increasing over time as the high mano species decrease. So that relative percentage between the two kind of shifts much more towards the mature glycans by the end of the bioreactor run. So this has really led to a better understanding of our cellular processes and how we can try to modify those, what levers we can pull, et cetera, to modify that production. So just want to summarize by, again, highlighting the importance of the optimization of your native SEC parameters, looking at different buffer conditions, looking at your source parameters, your ion optics, and of course, your mass analyzer is really going to have a lot of benefit. That allowed us to lyse cells under the non-denaturing conditions again to maintain our intracellular aggregation and compare that to our extracellular aggregation, which then led to a follow-up study looking at how the immature mannose glycans are changing and really highlighting our dynamics between the endoplasmic reticulum and the Golgi apparatus. Some limitations of this and things we would like to address are looking at possibly using a subcellular fractionation method so we can investigate, glycans in the ER versus the Golgi apparatus and kind of see if we can tease apart enzymatic processes, bottlenecks, et cetera. Additionally, we would like to couple our native SEC method with a denaturing SEC method so we can start to get a little bit more information on the exact nature of those aggregate species. Lastly, I just want to acknowledge our director, Partha, and our analytical sciences team that contributed to this, Hirsch, Andrew, and Elsa, and then also our cell engineering and cell line development team that whose work went into this, Tom, Lauren, and Kate. And with that, I'm happy to take any questions, or in the interest of time, if we have to share those, save those for later, I'm happy to.
Yeah,
Again, thank you for your time.
We'll hold them to later. But if anybody has a question, put it in the chat. I'm doing my best to monitor, and we'll see how well that goes when time comes. So next up, we have Francois, who will tell us about AIDA, Accelerate, top, middle down MS data analysis, application to deamination, isomerization, and oxidation in biopharmaceuticals.
Okay, do you see my screen?
Yep, looks good.
Okay.
You're good to go.
Thank you. So thank you very much for the opportunity to present today how we accelerate top and middle down MS data analysis using all ion differential analysis, or in short IDEA, and how we apply it to the characterization of biopharmaceuticals. Let me switch. Okay. Just a disclaimer. So these slides are for this particular workshop. So please contact me if you want to distribute further or if you have any questions. So I'm heading a mass spec group within Novartis at the Technical Research and Development Biologics. We are based in Basel in Switzerland, and we develop different type of biologics, antibody-based, multispecifics, are also non-MAbs based. And we perform product characterization from pre-IND to post-biologics license application. And our bread and butter task is basically to characterize our critical quality attributes of these products and to build the product and process understanding to ensure that we manufacture safe and efficacious products. And of course, mass spec is really the cornerstone technology to characterize and monitor these CQAs. and we use the peptide-based multi-attribute method for this task. So we also apply intact analysis, subunit analysis as part of our routine packages, and fragmentation is done also to provide fast complementary mass and sequence information during early method development, early peak characterization, but also later in the process when we perform impurity characterization. So we know all these different flavors of MS approaches for variant characterization, and which leads to different kind of complexity of spectra. And when we applied, I think this is maybe on top of, when you apply middle down analysis 10 years ago, it was on the thermo-orbit rapelite, which is now retired. It had different fragmentation modes, and we had different challenges at the time. So here it's an example of a Sigma map fabricator digest where we separated different subunits in the control and hydrogen peroxide stress samples to induce oxidation hotspots. And when we looked into this fragmentation map for both the variants but also the subunits, it was very time consuming, especially to compile all the evidence with the different fragmentation mode. On top of that, this process may also lead to ambiguous localization of PTMs, especially in chimeric spectra. And diagnostic ions may also be missed if there is mass computation error during the convolution. So we wanted to see the data at the time, and overlooked fragmentation patterns. So this may not be assessed at the time, and this was also pretty much focusing on terminal ions. While actually after a few days, you could also conclude that your experiment was inconclusive because your modification was in non-fragmented regions. That's why at that time we came up with the idea to use all ion differential analysis. So basically to reveal first diagnostic differences between variants before any kind of assignments of fragments. And when I mention all ions, I mean terminal fragments and internal fragments. I don't know if you see my mouse, but maybe I can use a pointer. Yeah, it's better. And also consider fragmentation efficiency if we consider differences of fragmentation. So we established some workflows within gene data expressionist software to compare different MS2 spectra together and to perform peak detection, isotopic peak clustering, and monoisotopic mass computation together. And then we apply different statistical tools, which in a few 20 minutes, you would basically find your diagnostic ions. Yeah. Here we have scatter plot analysis, presence of new peaks, but also volcano plots, for example, if you run with replicates. And then after finding these diagnostic ions, then you look into the fragmentation, the fragment assignment, depending on your fragmentation type, modification, but also potentially looking into internal fragments. And here we consider the N-terminal to proline internal fragments as good solid hits before validation of ions with isotopic distribution and abundance. A bit of ideation. So of course we were influenced by pioneering works of people in this field, focusing on increasing sequence coverage, but also people working on oxidation and partial sequence interpretation already 15 years ago. While other groups were focusing on the study of internal fragments, especially on small proteins. Others also identify that most of the ions were actually not analyzed at the time in some middle down proteomics data, and they were basically linked to internal fragments. Well, of course, fragmentation efficiency has been studied for a long time. Fragmentation efficiency, but also cleavage properties of intact protein bonds. Some excellent work also was published with data repositories about cleavage property. And we use this work to combine all these elements to focus with idea on deamidation, sequence variant, and oxidation. And our results at that time was to 2019 publication, where we demonstrated that we could also increase the sequence coverage by up to 30% with internal fragments and terminal to proline, and we validated this confirmation, this study with sequence variant. And more recently, we studied multiple isomerization variants and the isoaspartic acid effect. And in the meantime, we were very happy as well that other groups and especially academics were focusing on the study of internal fragments and also showing that there is about 30 to 40% sequence coverage increase. So if I go to the example of the Sigma map fabricator digest, where you have a control sample and stressed sample with hydrogen peroxide, and here we focus on one glycoform. You have here, for example, a mono oxidation or double oxidation, and here actually you have multiple species coeluting. And when you fragment, you actually again process all your spectra together. So you have basically your mono oxidation species, but also your doubly oxidation species. And with the scatter plot analysis, you can really identify what are your differences, synchronize the views with the raw data, and really look at the difference. So mapping terminal ions, monitoring basically the oxidation at multiple sites, but also finding some potential internal fragments. So as mentioned, we focused also in this study with Sigma MAB to showcase that internal fragments and terminal to proline could be used to increase sequence coverage by 30%. And here this is a view which was adapted from this paper, where we showcase the average number of internal fragments per motif, where we have actually a non-even distribution, so very targeted fragmentation depending on the motif, but also sequence environment. We further validated this finding with a specific proline to histidine sequence variant, in which in this very similar spectra you could actually spot some missing proline and terminal to proline 59 internal fragments. I think I will switch to deamidation now. And I think I've also heard earlier that it's important to have separation of your variants online. So here we always focus on also the chromatographic separation of the variants. So this is very key, especially for deamidation. And here you have your deamidation variant and your light chain. And here you can clearly see the shift of isotopic distribution. And when we perform HED fragmentation, so you have a very similar, of course, fragmentation pattern. So here that's all your MS2 fragments, but actually there is a population of ions which are twofold increase. These are these ions which are a bit higher in the deamidated spectrum, and they are all consecutive to the deamidation side. And this twofold increase was also showcased in this Avalant and Nilkeller data repository of fragment propensity for the LD compared to the LN motif. And of course, we could also confirm the shift in mass for terminal and internal fragments. For ETD fragmentation, again, very similar comparable spectra. And here, actually, we have an opposite phenomenon where we have at the site of deamidation actually a reduced fragmentation. And here we hypothesize that based on the introduction of the negative charge of the aspartic acid, then we also repel the ETD fluorine 10 reagent, just impacting the fragmentation. And I will finish with the example of isomerization. So here we also work with the light chain and separation of isomers, so again key. And here we use a very stressed material to enrich different levels of isomers in peak 1, 2, and 5, which are showcased here. So the same isotopic distribution. And again, very similar fragmentation spectra acquired on the Lumos instrument. And in a few minutes, you can really plot with your replicate analysis the significant changes, which were with your specific diagnostic ions, C90 plus 57, but also the C ion N-terminal to ISO-D, which we consider is the ISO-D effect, ISO-asp effect. We also did some molecular simulations to consider changes at specific site of a CDR loop, and we could investigate the different fragmentation pattern which were induced by these different modification. And because of specific heterogeneous bonding and different fragmentation site, we hypothesize also stabilization or increased stabilization of some modifications such as the ISO-D, and this could explain also some isomerization at unpredicted bulky site like DW and DF motifs. Then I will conclude. So I hope I could convince you that a top-down, middle-down MS workflow can provide fast complementary mass and sequence information. So especially if for peptide mapping you have actually small or very large peptide where you miss actually diagnostic ions, here the IDA approach can be very beneficial for mapping oxidation, deamidation, and isomerization. While the idea is can speed up analysis across multiple variants, multiple replicates, and leverage data from different fragmentation patterns. And I would like to celebrate the teamwork, the collaborators who basically also supported this work, and which was also part of the JASM cover in May last year, and also collaborator at Gene Data and the support. And this work will also be presented in more detail at the Gene Data Partner Symposium in March in Basel, Switzerland. So, and thank you very much for your attention.
Thank you very much, Francois. I think it is. So now we will transition from the wonderful talks that we heard, and as places to spark conversation also in the panel's discussion here. So if you have a question that you want to ask of anyone, please feel free to raise your hand, and we will have you unmic and ask your question. That is my preference, by the way. I don't want to just be reading questions from the chat. And thanks to our speakers for addressing some of the questions in the chat, but it's maybe hard for me to track exactly. So, you know, if you have questions, raise your hand. I did capture some of the questions from the chat, so I have those I will ask also. But I'm gonna ask the first question as people are thinking. So it's interesting, right? As I spend a lot of time thinking about analytical characterization of biopharmaceuticals, whether that's mAbs, more complicated antibody products, cell and gene therapies, whatever it might be. And as these new technologies and applications come online and help provide additional information to the process, I'm wondering if you have any insight from yourselves or others in your companies or entities on what regulatory challenges you might expect in trying to implement and include these in regulatory submission practices. Regulatory submissions and practices. That can be for anyone before I just call on someone.
Maybe I can start and just offer a brief answer. So I think one of the complications is actually choosing which mass spec platform and data processing software you put into the GMP space. As you know, I think a lot of us know, you can get different, slightly different results based on the instrument that you're using. And I think the workflows are also still being developed in that space for like GMP applications. So we run into some issues, of like different desolvation and even the distance like between your tip to the instrument. And then of course, like I mentioned, the different sources. So that's definitely a challenge, and making sure that, if you do put it in the GMP space, then your other collaborators can use that same instrument, or at least know how to transfer your method to their instrument for use to get the same reproducible data.
Yeah, that's actually a really good point. I mean, it's actually interesting as I think of, as well, you talked about MAM, and I think about more traditional MAM and how that needs to be, in my opinion, implemented differently in the process, and whether and in the GMP space, whether that's at line, inline, or online, really thinking about process analytical technology, real-time lot release, continuous manufacturing, all those kind of things as part of the equation is the analytical chemists that we are have to think about those things. And one of the things that I often think about also is, if you think about the product life cycle from easy in a very simple way, research, process development, and commercialization, I like to see the same instrumentation move from the research space all the way through the commercialization space. But in some sense, right, maybe that doesn't mean, and especially with native top down or intact or whatever, you don't need to worry about MSMS. So the instrumentation could be perhaps simpler because one of the concerns too, not only is instrumentation in my experience, but operator, right? Like we all might be mass spectrometrists, but the people who are working in the GMP space, generally speaking, are not, which is why they get nervous. But any other thoughts from anyone on this? Then I'm going to just run through the chat questions as people think of other questions I want to ask to raise their hand. The audience is very shy today. By the way, to all of you who presented, I'm going to ask you questions that were in the chat that I know you've addressed, but I think it's useful to pose them live so that you can address them for everybody who's listening and not monitoring the chat closely. So we'll roll back to Jack to the first talk, and I can only see some of you on my screen. So if you're not here, just okay. Could you do your experiments with capillary electrophoresis, like ICIEF in industry, maybe having better separation and peak capacity? By the way, I'm going to ask you questions from the chat, and I'm going to remove all the niceties. So just assume every question I ask from the chat starts with great talk.
So on CIF, a number of years ago, explored that, but from basically isoelectric focusing coupled to mass spec technologies that I'm aware of require a pretty concentrated sample. And so we immunocapture from serum. So our sample is not that concentrated. So we essentially need to be able to put inject as much volume as possible from that sample. So, ideally, if we elute, let's say after immunocapture, we're eluting and we have 100 microliters, we might look to inject, if our loop is large enough, 40 or 50 microliters to get as much of that sample on column as possible. So with isoelectric focusing and the volumes involved, I'm not aware. It doesn't mean the technology maybe is not out there, but I'm not aware of any of the technology that would allow us to load that much. So we'd be stuck loading a few microliters, and then overall, the sensitivity wouldn't be as good. And we did try this in the hands of a demo we did with a vendor that had a commercialized isoelectric focused mass spec platform, and the performance compared to LC was just not even close. So now maybe there's custom setups and maybe new technologies, but that's the rationale for LC over IF.
Perfect. Yeah, makes a lot of sense. Quick question for Quarantine. What is the amount of antibody you used to acquire the LCMS top-down data?
Yeah, I saw the question. So yeah, we were loading 30 micrograms of MAD on the column usually. So yeah.
Juan, I saw you answer this. Some of this in the chat, but would love to have you articulate that. Can you comment on how you distinguish true in-source fragmentation from declustering and desolvation in native, intact, and mass spec.
Yeah, I guess we would just, fragmentation that's happening during the ionization process or transport of the ions prior to the mass analyzer, we can't really distinguish those unless we set up like an isolation event in the quadrupole. Would be one way to do it.
Makes sense. All right, Shannon, how do you determine your VPs ratio using peptide mapping? And is it using peptide mapping and terminal peptides? Do you have a specific set of peptides you look at, etc.?
Yeah. So I think this, the VP ratio is definitely one of the CQAs that is challenging for AAVs because you have those overlapping N-terminal sequences for your VP2 and your VP3. So there are a couple tricks that we are able to play in order to use different assays to like orthogonally characterize the VP ratio. Analytically, we use CE, but that's not we're not using that as a mass spec technique. It's CESCS, and we can quantify the different VP ratios. But for a multi-attribute type method, we're starting to use peptide map. So here, the different sequences, the N-terminal sequences of 1, 2, and 3 have unique characteristics. So the VP1 is acetylated at the N-terminus. That one's also the easiest because there's no other overlap between 2 and 3. And then VP2, it has overlap with the VP1 sequence, but the first threonine on that is actually truncated for VP2 proteins. So you can look at the signals between the truncated peptide versus the untruncated and calculate your VP ratio for that one. And then for VP3, in a similar way, that N-terminus is acetylated and truncated. So you have some different properties that you can look for in the peptide map and then compare it to the native sequences that overlap from VP1 and 2. Hopefully that made sense.
Talking. Yeah,
maybe
that makes
for
sense.
my AAV folks.
No, it does. And while we have you here, I'm just curious too, as you characterize AAVs from an analytically, obviously, do you see any differences in, like not in my experience, and we do minimal characterization. We do a lot of expressions of AAV serotypes, and they don't express all the same, and they're quite challenging on occasion. Do you see any differences when you look at different serotypes?
Yeah, actually we do. And I think that's something of interest in the field as well. So from a CDMS perspective, from like an intact capsid, the empty partial and fulls for different serotypes seem to separate better or worse depending on the serotype. So I think that maybe has to do with the serotypes packaging full or maybe fragmented portions of the genome differently. So that's definitely one difference we see. And in terms of like post-translational modifications, we don't see too many differences between the serotypes, but we do see differences between empty and full. So some cryo-EM images have shown that if you have a genome inside, then it actually changes the conformation of your capsid a little bit. And that can make some, for example, deamidation sites more susceptible to deamidation. So we do see some of those differences, but not too much in terms of serotype for the PTMs.
Yeah, that makes sense. And before I switch off, I'm going to ask my cranky questions to you
no.
about AAVs in particular. So, and I travel all the product classes, so I'm an expert in none and an expert in all at the same time. It fundamentally makes AAVs are fundamentally toxic, and I wonder if we all, as an industry, which includes academia, are spending too much time on AAVs as a delivery method, as opposed to thinking about and really investing more time and resources in less toxic delivery methods.
Yeah, I think there's been a lot of data recently showing why some of these molecules are toxic. So I know some companies are mutating regions of the AAV, and we've, as an industry, found that some of those mutations do cause liver toxicity. And I think that, those different engineered capsids are kind of what's booming in the news right now. But in general, I think AAVs are very well tolerated and they have a very low immunogenicity. And that's why, they have been used for so long for delivery vehicles. But another point, we're starting to change how we administer these molecules. So if you administer them intravenously, you have a lot less control. I mean, the serotype does help traffic them, but you do have a lot less control of where they get trafficked in the body. And sometimes different serotypes can end up in your liver and cause liver toxicity. How we're starting to circumvent that is injecting directly to regions of the body that we're trying to treat. So we just acquired a company that directly injects AAV medicine into the cochlear of the ear. And this has been shown to completely regain hearing in children with OTOF-mediated hearing loss. So I think some of it is still a little bit unknown, and I do agree that we should move forward cautiously, but some of these medicines are just completely changing people's lives, and I think that's really important.
Yeah, I agree. I mean, I will tell you that when I teach, which is not as much as that used to be, I talk to the students about how when I was in college and going through grad school, we were, biologics weren't new, but they were relatively new-ish as a therapeutic. And we were still talking about treatments. And as they move forward and we move into these new spaces with cell and gene therapies or other oligotherapies, we're moving towards cures as opposed to just routine treatment. So an exciting time for sure, which then pivots me to BFAN. A couple of questions about desalting offline with a zip tip and then not linked, but at the same time, how many different RNA PTMs were you able to identify using your method?
Yeah, I can articulate the answers a little bit more. So regarding the offline desalting, the purpose of this column base is really to do everything online. That the pH of the 20 millimolar piperidine, it's about 11, which is quite high. But if you do it on an online fashion, in our data, we did not see any degradation due to that. Doing it offline do have potential risks of having degradation. And because it's an SEC mechanism, you are not retaining anything or doing any separation. So finding out what fraction or what time frame to collect from a zip tip type or SEC spin column type can be challenging. So you would probably need to do some development there. In theory, it's probably doable. Regarding the modification, so in this case, synthetically made guide RNA, we are seeing impurities, either from it's mostly from the solid state chemistry, like phosphorothioate PSPO, and during oxidation, reduction, leaving group cleavage, stuff like that. We did see modifications on that. Given the removal of adducts, I can imagine for other application, other oligonucleotide modifications on it, you should be able to see it as well. The real challenge for impurity in this space is a lot of the impurities that you're seeing can happen in any residue, but on MS1, it's heterogeneous, but you do see as one peak. So that's a challenge that's hard to resolve by mass spec alone. Even if there's additional truncation, it could happen in anywhere during the sequence, and that doesn't show up as different peaks on mass spec. Yep, I hope that answers the questions.
Great, thank you. I'm just going to transition to more broad questions for all of you. So what is the overall, this is from the chat also, what is the overall status of native, intact, and top-down MS in your respective companies? Is it a cool tech to investigate something that is sometimes useful in discovery, or a mission critical tool, or something in between? How do you see it going through? How do you see it playing in the lifecycle management of a product? And in that same context, what is the next iterative improvement that you need or we all need for native mass spec? And anyone can start, but I would like each of you to address that with your thoughts.
Maybe I can start. So what again in my lab, so we use this middle down analysis specifically for peak characterization. So when we have a new method, and we can apply this as someone also mentioned. So when you want to do this with peptide mapping, sometimes you lose the actual information, while when you do this with middle down analysis, actually can retain this information on subunit. And one example I mentioned at the end of the talk is sometimes you have with peptide mapping a very short peptides, so the amylation on short peptides, or isomerization on long peptides where you have multiple sites on the same peptide. And sometimes the diagnostic fragmentation at peptide level is really minimal, especially if you look at very low levels of these modifications. And again, when you want to have complementary information, it's very orthogonal and also can bring actually the site localization if you have really better diagnostic behavior. I'm thinking also when you have a modification which is found only in subunit analysis, but actually it's lost in peptide mapping because the peptide is so small, then it's also very interesting to get directly the fragmentation from your subunit analysis. So I would say that to answer the question, it's not only cool, it's useful also in characterization, whether it's in early or late.
Yeah, I can follow up on Francois' answer. I do agree that that's one of the really good points of the middle down and top down, is that you can really link the modification on the site of the modification with the intact molecule. That's the information you're losing with when doing the peptide mapping. So that's really something interesting, and that can be useful at all the stage of the analysis. And it's also a bit less time-consuming and labor-intensive, let's say, in terms of sample prep, at least. Data processing might be another story, but for the sample prep, at least, to get direct information on your sample.
So to speak to the point on kind of the status of native intact top-down, so from discovery bioanalysis, which is where I sit in the organization, we're looking to understand the performance of a biomolecule in life with the aim of taking it to the clinic. So it's critical to have lots of tools in the toolbox. Intact mass spec is one of those tools. And I think it's really key to be able to have a number of sharp tools to use with different types of molecules and answer different questions and be efficient in that process. So to me, there's lots of aspects for top-down intact mass spec that can be sharper and quicker than digestion-based methods when you need to use that tool. And so I think it'll continue to grow and prove advantageous.
In our field where we're at with Johnson & Johnson, I'm more or less in the cell line development group, so we're kind of at that nexus of discovery to manufacturing and scale up. So we're not necessarily trying to characterize a lot of drugs, but trying to characterize the same drug across a lot of different samples. And that has really made native an absolutely critical assay for us in terms of looking at different aggregate species and things like that, really getting a read on not just purity, but what are the impurities. And native mass spec has been very difficult to replace in that sense. We've also coupled it with, say, like subunit digest and things like that. So it's kind of getting closer to that middle down. But one thing that we have not really implemented is that traditional top down where you're actually fragmenting, trying to get sequence coverage, trying to get PTM analyses and things like that. And that's something that I think is still in the, I guess, cool stage for us, where it's not really platform. It's not an everyday analysis, but something that we're like, oh, that would be nice if we could do that. It is unfortunately a lot more difficult. There's fewer people doing it. There's a lot more, fewer resources for it. So that's something that I think we would like to build on, is incorporating that. And then also I think the other question was like, what would be sort of an iterative improvement? I think would be sort of a front end modality such as like ion mobility or something like that to layer on top in a very like easy to use fashion. I think that would give us a lot of information when we start to see a chromatographic separation, but we don't necessarily have a different mass spec reading, and kind of wondering, is that, you know, we can hypothesize all we want that that is a conformational change, things like that, but without that an actual assay to back that up, it's difficult to make that assertion. And I think that's something that I would like to see very, maybe not easily layered in, but in a feasible manner.
Yeah, I just echo what Christopher said there. So native MS, getting intact molecular masses for ADCs in the development space is just absolutely critical. So it's a tool that we use routinely. But when you get to like fragmenting those molecules using top down, middle down, that is more like cool applications. It's something that could potentially be useful, but not applied routinely yet, just due to like the difficulty. So I'd say things like top down, middle down are used on a case by case basis, where it's like there's no other easy way to get at that information. But getting site specific post translational modification information, that's still going to be in the wheelhouse of peptide mapping.
Yeah, I totally agree with what was said here. So at Lilly, I think our mass spec group is pretty well integrated into the entire drug development process. So we still utilize like intact mass for things like peak ID for our analytical group, and characterization of like others talked about here, like the DAR and the intact molecular weight, which is an FDA requirement for IND and BLA submissions. But I think we're kind of at a very exciting time where we're using more and more top down in the group, because these other techniques really can't get at some of the information that we need. So right now, as others have said, it is kind of a cooler thing that we're working on. But I think moving into these newer modalities, it's going to be necessary to completely elucidate the structure of the molecules and help inform the analytical control strategy moving forward. And then I also wanted to touch on, the multi-attribute method as we move more into that space as well, using mass spec into regulatory spaces. I don't think that, top down and native are really going to play too big of an impact, but the intact mass is definitely something that we're looking at moving forward for the MAM applications. And then kind of to make the process easier, I talked about this a little bit, but just standardizing a lot of the workflows I think is still necessary in order to completely implement it into our workflows, especially GMP. So, better separation beforehand or better integration with mass spec. Because everyone still loves LC, right? So I don't think we're going to get rid of it anytime soon. So better implementation of LCMS integration, I think, is good.
I don't love LC, by the way. If we could get rid of that and just go to the mass spec, I'm game.
Agreed.
So everyone had a chance to share their thoughts on that question. And I actually also agree. We do a lot of thinking actually about intact mass in in the product lifecycle as a characterization tool. But also, like I tell my students who come to work with me because I'm a mass spectrometrist, that you don't need the most expensive analytical tool all the time to answer the question. Don't need to hit the nail over the head with a sledgehammer when all you need is a traditional hammer, which is part of what we think about also. Are there other questions that folks have who are here in the audience, or want to put it in the chat? Raise your hand. Everybody is super quiet today. That people want to ask about. Any questions anyone has? I mean, I think the other thing that's interesting that I think you mentioned, Shannon, and I think that this that I do a lot of thinking about these days because I am an academic and they make me think about it at the university, is really the I've always argued that large molecule, hardware always is better than the software. And really the software applications are good, but could be great. And now with the sort of integration of AI and I think that we're also at a really interesting time, because my perspective is with AI, it's data in, garbage out, just like I tell people, garbage in my mass spec, you're going to get garbage out. But the data quality that we saw today is and others produce, obviously, really is prime time to think about new analytical bioinformatics, for the lack of a better term, tools, and AI integration. I mean, are all of your groups thinking about AI integration into analytical workflows, and how are you thinking about that? If you are, that's for anyone. Nobody's thinking about it. Quickly, you better start, because the world's moving there.
If I may, so of course, I think every company is considering AI, let's say, in development. So, but here in terms of software, of course, let's say the MS software provider are also considering AI, and I think. I mean, I don't want to say a few names of company, but of course, to integrate this to get a faster response. I think you need. I mean, as mentioned, sometimes you just need a good database already, because before implementing AI, so finding, leveraging the good data, and AI may be in the end also biased if, let's say, there is too much data which are conflicting together. So it's a bit like chimeric spectrum. if you end up with things which are in opposite direction, that may be not so good. So I guess the AI will need to leverage data from a very good, very well curated database, from scientists, from consortium, from things. Otherwise, it will be generated in crap. Yeah.
Yeah, definitely want to echo, of course. Yeah, I think everyone is thinking about it. One challenge that we've had as a department and in development is handling all of the data that we have, right? So yeah. I don't think any of us have a problem generating a lot of data, big data. The problem is handling it. So we are trying to make our lives easier by building tools to search, previous data sets and outcomes from previous modalities, so that once we do, kind of establish ourselves in a modality like ADCs or AAVs, we can look back on historical data and say, oh, this didn't work with this cell line, or we had a problem with this sequence in this previous molecule, so let's try to avoid that for future. So I think that's a subtle bit of AI that, well, maybe it's not really AI, but data, integrity and housing and searching is like the bane of my existence right now. So we're getting better, and I think with the better software tools that are available is helping. But that's our current biggest endeavor, is implementing that so that we can move really quickly with these modalities that we're establishing.
Sure. Paul, I see you have your hand up.
Yes, I do. First, thanks for everyone. It's fantastic. I really enjoyed this. Just a question from the consortium point of view. A lot of you mentioned, standardization, difficulty to submit this kind of data, and you all have very fascinating but different workflows. Is there anything the consortium can do to try to bring some coherence to any of this that would be beneficial to your organizations? We've done things in the past where we've tried to run, develop standard languages, develop some other guidelines around protocols. Is there anything we could look to do from a consortium point of view to help some of you in moving these things forward? And if the answer is no, that's fine too. We're just curious and kind of open
I mean,
to.
I would say hosting events like this is helpful, just to, create a space where people from industry can come together and share the different projects that they're working on, and then have open discussion, and we can see where the challenges are, and sort of where the status of the technology is, and then, helps build consensus going forward, and just normalizes it more and gets it more accepted. So then I think that pushes the use of mass spec forward with drug development as a whole.
Yeah, I would like to echo the same idea. I think that the barrier of getting into top-down and doing top-down has been a lot lower compared to years before, thanks to all the efforts that the consortium did. Having people talking about their application and sharing how they use top-down in their own work, it's beneficial for others to see the value of doing it.
Yeah, we can look to do these types of events in the future, maybe on a more targeted basis on certain subset of applications or things like this. We could definitely do those kinds of things going forward. And if possible, we could bring people in from the regulatory side if they were interested in learning more about it, if that would be helpful.
Yeah, I think the more exposure regulatory has to mass spec, the better. So that would be good.
I spend all of my time traveling the world trying to help them understand what mass spec is and how it can be useful in their processes, in their analysis of their data packages, historically in bottom-up applications, but have recently started talking more about top-down and impact and native mass spec applications. So, they're a little they're a little gun shy, for lack of a better expression, but I think that's part of the reason why I get to be an academic and try to do those things as well. Because if we don't want innovation that you all work on to be the roadblock to getting medicines to patients who need them. All right, if there are no other questions, I think we'll wrap it up a little early today, give you all some time back. But again, I want to echo my thanks to all the speakers and to all of you for being with us today. Super interesting topic to talk about, top-down impact and native in biopharmaceuticals, something near and dear to my heart. And I look forward to seeing all of your continued work, but also a greater uptake of those technologies throughout the product lifecycle. And just as a reminder, everything today was recorded and will be made available through the top-down consortium. And we look forward to seeing you at the next event.